This is Navi and Emma's first presentation on the case study Which Little Piggy Went to Market, Bioinformatics, and Meat Science. So what actually is bioinformatics? Well, bioinformatics is the use of computers, mathematical tools, and statistical techniques to analyze biological information. And what it allows us to do is it allows us to examine genetic data like DNA sequences. Let's go ahead and identify the problem in this case study. So, a meat distributor or a butcher named John suspects that his distributor is providing him with sow meat that is tainted with boar meat. And so John asks his friend Sue if she can help him find a way to test the meat to see if it contains traces of boar meat. So, basic information that we need to know is that a sow is a female pig and a boar is a male pig. And the meat that boars contains androstenone and skatole, which are hormones that accumulate in male pigs who have not been castrated. And what this does is it results in their meat smelling of urine, feces, and sweat. Question 7 asked us to design an experiment that will show the presence of boar meat in a mixed sample, keeping in mind that biochemical tests are too slow and relatively insensitive. So what do we have to do? So in this case study, we will be examining genetic data, DNA sequences, or also known as DNA sequences, in order to determine if the meat is tainted. So first we asked ourselves, what is the difference between males and females? So we know that males have the XY chromosomes and females have the XX chromosomes, but what gene is found on the Y chromosome that is not found on the X chromosome? And so the answer is the SRY gene. And the SRY gene contributes to male characteristics, and it is only found on the Y chromosome. So our chosen method of experimentation is polymerase chain reaction, also known as PCR, and PCR amplifies the DNA of a specific sequence. And so in this specific experiment, we will amplify a DNA sequence containing the SRY gene, which is located on the Y chromosome, in order to determine if a sample contains boar meat or not. In order for us to successfully complete this PCR experiment, we will need all of the required materials listed below. So we will firstly need 2 different primers, which are complementary to sequences at the end of the DNA region, which we are amplifying. And so the DNA region we chose was from 3,901 base pairs through 4,291 base pairs, and both our forward and reverse primer are 20 base pairs long. And we will also need DNTPs, which are basically nucleotide building blocks, and they stand for deoxynucleotide trisephosphates, and they consist of DATP, DTTP, DCTP, and DGTP. And we will also need Taq polymerase, which is a heat-stable form of DNA polymerase. And Taq polymerase synthesizes DNA from our nucleotide building blocks, or our DNTPs. And we will also need DNA, as it will serve as our template for the entire experiment. We're also having a variety of controls and test samples in our experiment. So our positive control 1 would be a normal male boar, being SRY positive, and our negative control 2 would be a normal female, being SRY negative. Negative control 3 would be a sample without any DNA at all. Our negative control 4 would be boar's meat, but contain no primers. And lastly, our test sample will be the unknown, so it might have a mix of boar meat and a mix of sow meat. We're unsure of that yet. And our primers will be used to target the SRY gene. So before we actually begin the process of PCR, we have to first extract the DNA of each of our samples by bursting open their cells. And then we mix our DNA samples with the specific primers, except for negative control 4, the DNTPs, and also Taq polymerase. So now that we've successfully created our samples, we are going to proceed through the steps of PCR. The first of which is called denaturation, and this occurs at a temperature of around 94 degrees Celsius. And so In this step, we apply heat to the double-stranded template DNA in order to separate its strands. The second step is called primer annealing, and this occurs at around 60 degrees Celsius. It can be lower, it varies depending on the primer. So it could be lower, it could be closer to 50 or 55. It could also go up to 65, but that all depends. And so in primer annealing is when primers bind to the specific sequences on the DNA template as the temperature is lowered. And the third step is called primer extension, and this occurs at around 72 degrees Celsius. And primer extension is when the temperature is raised slightly, and Taq polymerase uses DNTPs to catalyze the synthesis of complementary DNA strands. So as a result, the length of the primers is extended by the synthesis of DNA. And so we're going to repeat these 3 steps for 30 cycles, and so at the end, when it's all complete, we're going to have 2 to the 30th power amount of DNA that we started with. So what that means is we're basically going to increase our DNA about 1 billion-fold. So that's how effective PCR is at amplifying DNA. So now that we've amplified the DNA for all of our samples, we're going to move on to the second part of our experiment, which consists of gel electrophoresis. And gel electrophoresis is basically allows us to separate macromolecules, for example DNA and proteins, based on their size, length, mass, and charge. So the first step in gel electrophoresis is that we're going to set up our apparatus, which we can see here in this photo, and we're going to create a TAE buffer, an agarose gel as well. And so once we pour in the gel and it solidifies, we will remove the comb and then add the TAE buffer to the apparatus. And so we're going to take our positive control 1, which is the male DNA of the boar, and we're going to pour that into well 1. Well 2, we will pour our negative control 2, which is female DNA, so the sow. Negative control 3, which contains no DNA, is going to be poured into well 3. And negative control 4, which contains boar DNA but no primers, is going to be poured into well 4. And we're going to pour our unknown sample, or a test sample, which we don't know if it contains sow or boar or both. We're going to pour that into well 5, and then we're going to start the gel electrophoresis and let it run for 15 minutes, give or take. And so once it runs for a set amount of time, we're going to observe our results under a black light, and so we can then read our gel and the bands present. And so we're also going to measure and pour equal amounts of each of our solutions into each well, and we can't forget to add the loading dye to each sample as well, or else we won't be able to see the band. So those are very important steps as well. And here are our predictions if all goes well with our experiment. So the male boar DNA would produce a band indicating that DNA was amplified because, yes, the male boar contains the SRY gene, and it's present. The female sow DNA would not produce a band because no DNA replication would have occurred due to the primers having nowhere to bind, because females do not possess the SRY gene. Remember, females only have 2 X chromosomes. They do not have the Y chromosome. And so our sample without any DNA will not produce a band as well, because without DNA, no replication can occur, no amplification can occur, obviously. And the boar DNA with no primers will not produce a band either, because the primers are necessary for PCR. We need primers to bind to the DNA sequence in order to amplify them. So here they were absent, therefore no band would be produced. And so if the test sample does indeed produce a band, then we can conclude that yes, boar meat is present in it. And if it does not produce a band, then we can conclude that it is from sow. And so that is our experiment and answer for question number 7.